I went to medical school in the Netherlands. I started in 2007, and in Europe, it's a continuous program. You go to medical school right after high school. I started that, and towards the end of my medical studies, I wanted to get some research experience, and it was actually also part of the curriculum that everybody in medical school in the Netherlands has to get a certain number of research credits. And so I spent some time in the laboratory of R00 Pathway in MD Anderson in Texas at the time. And that's really when I started getting into research. And when my internship was completed, R00 was like, well, why don't you stay with me for a couple more months? And fast forward 7 years, really, and I'm still here. So my work really centers around the study of glioma, which is a type of brain tumor. And most brain tumors are not malignant, but glioblastoma and gliomas are some that are, and they're unfortunately quite common, and we still don't have good treatments for them. And so we use genomics to study this disease, and by that I mean we look at the DNA of these tumors. Subtypes of glioma were very specifically enriched in a certain type of telomere maintenance, and telomeres are the ends of chromosomes, and they shorten with cell divisions. And so, and cancer cells, because these cancer cells proliferate and replicate so quickly and so rapidly, so they lose a lot of telomeres, and this is detrimental to the cell, because with each cell division you lose a little bit, and so we need a mechanism to replenish this DNA. And so there's really two main mechanisms. There's telomerase based, and there's the alternative lengthening of telomeres mechanism. And so we noticed that, well, this oligodendroglioma subtype had often TERT promoter mutation, was telomerase driven, as were the glioblastomas. But then this astrocytoma IDH mutant subtype of glioma was showing this alternative lengthening of telomere maintenance, which is recombination driven. And so that kind of got me really interested into this topic of telomere biology, and that kind of segues into the topic of my K99. So the title of my presentation was the genetic and epigenetic strategies for the acquisition of telomere maintenance. As I mentioned, telomere maintenance, we noticed these two different types were so prevalent in glioma and so contrasting between these two subtypes. And, I just became really fascinated by trying to figure out what it is that tumor cells have to do to kind of steer them down one pathway or the other, and really how that happens. And so, we started looking into how telomerase becomes reactivated. So there's TERT promoter mutations that are, common in one subtype, and the other subtype not as common, but they're still quite frequent. But we wonder, well, are there other mechanisms or other requirements to kind of allow telomerase to become active and TERT to become expressed, and that's kind of, in a nutshell, what I was really, what got me, what drove this proposal. I think I had a really strong publication track record with R00. When I started out in the lab, we started working on this molecular pathology of neuropathology of trying to understand, use genomics to study brain tumors, and so I had a co-first author paper in Cell. And then that kind of segued into my interest into studying telomere maintenance mechanisms. And so using genomics in the TCGA, we started trying to infer telomere maintenance from this gigantic 10,000 plus patient cohort. And so that paper was ultimately published in Nature Genetics. And so I had a first author Nature Genetics and a co-first author Cell paper. And then I also had a a review paper and several co-author papers. So I think that was extremely helpful. I chose was the National Cancer Institute, and so my research was cancer-based. And a really important factor was also that my PI was funded with his grants through the National Cancer Institute. So it seemed to make the most sense. Sponsored research administration team here, at the time led by Joanne Goodnight, was just fantastic. The science writers, Carmen, Dave, they were just incredibly helpful in preparing my application documents. Joanne herself, who led the Sponsored Research Administration, was actually at the NIH at one point where she was heavily involved with the K99 program, and so she was also helping me place calls to program officers, get in touch with the different institutes, and kind of connect with them about my aims page, try to figure out if my aims were something that they felt would fit within the institute and within something that they thought could be fundable. I can't thank them enough. I think I don't know if I could have done it without the support of the SRA team at JAX. The strongest points, I think, were my mentoring team that I assembled. I think the environment at JAX was thought by all the reviewers unanimously to be incredibly strong. I, as a candidate, was also received good scores. I think the most I got dinged for my actual research plan, the stuff that I wanted to do, which I think makes sense, and I think the reviewer comments were incredibly helpful, and if I had needed to resubmit it, then I would know exactly what to do, because it was really helpful. But fortunately, I got funded my first attempt, and so I didn't need to resubmit. Yep. Once we received the score and it became visible online, I contacted my program officer, who quickly responded that they thought I had a very fundable score, and then that I would very likely be funded. And so after that, it really took a couple months to kind of a pretty long time to really figure out what the budget was like and how the funding would look. But there was a couple of months. I don't remember exactly how much, but it's a long process. I got approached asking to apply to several institutes, like, hey, Floris, we noticed you have a K99. We have a position open. Would you be interested in applying? And so that helped a lot. And I think in general it's extremely helpful. I think it doesn't make or break your application in any way, but if you have one, it's definitely a plus. So starting my own laboratory, I'm very excited. I just accepted a position at TGen in Arizona, in Phoenix, and so I'm going to be starting there in August of this year, so that's incredibly exciting. I encourage people to apply. Start early. It's a lot of work. And don't underestimate the amount of work it takes to apply. Also start early, because you can't do this alone. You need a lot of people around you supporting you, writing letters for you, and so making sure that everybody's on board. It just takes a lot of time. So start as early as possible. And I'm absolutely available to people if they want to ask questions. Just shoot me an email. You can go to my website, www.barthel-lab.com, and take a look at the open positions. If you're interested, I'd love to have a chat with you, or coffee if you're in the area.